Hello guys, in this video we'll be reviewing the biology WAEC specimen for 2025. Alright, but before we go into that, if you haven't subscribed to this channel yet, please do so you don't miss out on any information that will be dropped here before your exam. Okay, so this first specimen is Specimen A, which is bean weevil. So what do we need to know about this specimen? Firstly, the scientific name. So the scientific name is Acanthocylidis obtectus. It is found under kingdom Animalia, phylum Arthropoda, and class Insecta. Another thing you need to know about this specimen is the feeding mechanism. It exhibits biting and chewing mechanism. Alright, and it also undergoes complete metamorphosis. That means the metamorphosis goes from the egg, the larva, the pupa, and the imago or adult stage. All right, now another thing you need to note is that the stage that burrows into the bean seed is the larval stage. So you may be asked, which stage of specimen A is responsible for burrowing into the bean seed? It is the larval stage. Okay, so the eggs are laid by the adults. The eggs develop into larva, from larva to pupa, and pupa to the adult, and that's how the cycle continues. You can also be asked to list the observable features of specimen A, and the first one here is that the specimen is divided into three parts or three segments, which is the head, the thorax, and the abdomen. Number two, it has three pairs of leg, or you can say it has three pairs of jointed appendages. Still the same thing. Three, it has there's a presence of compound eyes. Number four, it is typically or relatively small. Okay, so when you look at your specimen that will be provided before you, or that will be presented before you, you would find out that it is small. And most times when you want to observe, you have to use your hand lens when you want to observe the specimen. Now another one is that it is oval in shape, okay, and the color ranges from brown to black color. So these are observable features of the specimen. So in case you are asked to list the economic importance, these are the economic importance of specimen A. So it is both positive importance and the negative importance. So it causes severe damage to crop. That's one of the economic importance of this specimen. Two, it reduces the crop yield. It leads to food insecurity and scarcity. So when this specimen feasts on the bean seed, it can lead to scarcity. It can destroy the crop and lead to scarcity. Number four, infestation causes scarcity and escalation of prices of beans. So obviously when this weevil parasitize on beans, they destroy it, they reduce the crop yield, and that leads to escalation of prices. So when there is scarcity, there is escalation of prices. So these are the economic importance of specimen A. Other questions can include the relationship between specimen A and B. So what's the relationship between the bean weevil and the bean? The bean weevil parasitize on the bean seed. That's just the relationship. Okay, so without the weevils, you wouldn't see the holes. So specimen B is actually bean with holes. So those holes are caused by the boring of the larval form of the weevil. So the larval is what is actually responsible for boring, and it's the most active stage of these specimen A. And you can also be asked to draw specimen A. In case you are asked to draw specimen A, you should draw it and draw what you see. Draw what you see. Don't draw what is in your textbook. Don't draw what you can see in this video now. Draw what is before you. Label and title appropriately, very important. When you label, you title appropriately, and you write your magnification. Magnification is very important. How do you get that? You measure your specimen you have drawn on your book, and you measure your live specimen. Divide the values, and whatever you get, put it by the side of your diagram. That is your magnification. Also, you can ask the adaptive features of this specimen. It has hard exoskeleton, and it also has the chewing mouth parts. So I think that's all for specimen A. Specimen B is bean seed with holes. So the botanical name here is Phaseolus vulgaris. So that's the botanical name of beans. It is under kingdom Plantae. So what are the observable features? The most important one is that there is hole on the surface of the specimen. You can find powdery residues. You can find discoloration of the specimen as a result of the holes. You can also find weak texture. Now, how do you prevent the spoilage of specimen B? This question is likely to come out. So number one, you store in a cool, dry place to prevent mold growth. Yes, that's fungal growth. Store in a cool, dry place. Secondly, you store in airtight container to prevent insect infestation. Thirdly, avoid storing in porous or open bag. That's bags with pores. Avoid storing in such bags. Fourthly, control pests by using natural repellents like neem leaf or bay leaf. Not need, sorry. Neem, Neem, neem leaf or bay leaf. So that's the natural repellent you can use to control this pest. Check stored beans regularly for spoilage, so you can easily pick out the spoiled one, so you reduce the rate of infestation. Other questions can include, what type of germination does specimen B exhibit? So because it's a dicot plant, it will exhibit epigeo germination. Okay, that means the cotyledon is brought above the soil surface. Now, one part of the specimen stores food, obviously, is the cotyledon. Now, it has an annual life cycle. That means it completes its life cycle in one planting season. Specimen C, waterleaf plants. The whole. So the botanical name of waterleaf is Talinum triangulare. So that's the botanical name of waterleaf plants. Other kingdom Plantae and division Magnolophyta. Now, what are the characteristics you should look out for? It has flowers. It has cylindrical stem. It has shallow root system. And most importantly, if you look at the leaves, they possess pinnate venation. So pinnate venation is a type of venation that is under the web-like venation that is often characterized by dicot plants. So pinnate venation is what you can find in this specimen. You can be asked to draw a leaf from the specimen. So you pick out the leaf, draw, label. It has an entire margin. You can draw, label, put your magnification. Very important. You also need to know that this plant is a dicot plant. So that's that for that specimen. Specimen D is quill feather of domestic fowl. Now, what are the questions that can arise from this specimen? What is the kingdom and class from which specimen D can be obtained? So it is under kingdom Animalia, phylum Chordata, class Aves. And what's the location on the body of the organism? It's located around the wing region or the tail region. So an example of organism that this specimen is associated with is domestic fowl, pigeon, turkey, and the rest. And in case you are asked to draw, this is what the diagram should look like. These are the basic labels. the quills, the after shafts, the barbs, the shafts, and the veins. Now, after drawing this, you title your diagram, very important, and put your magnification. So that's that about specimen D. Specimen E is pineapple fruit. So longitudinal section of pineapple fruit. That means the pineapple will be cut from up to down. All right, so what's the botanical name? The botanical name is Ananas commusus. It's under kingdom Plantae and division Magnolophyta, just like the waterleaf plant. Now, what type of fruit is it? It is a multiple fruit, and it has inferior ovary. And if you are asked to draw, this is what your drawing should look like. You should capture the crown, the base of the crown, the ovary, the base of the fruit, the core, that's the inflorescence axis, the core, and the shell, maybe the shell, and that's it for that specimen. Specimen F is orange fruits, transverse section. That means it's going to be cut from left to right or right to left, anyone. And the botanical name of orange is Citrus sinensis. The kingdom is kingdom Plantae, division Tracheophyta. It has the axile placentation. That means the ovules are arranged in an axile placentation form. So the germination here is epigynous germination. You may be asked to draw. So this is what your drawing should look like. You have the epicap, the mesocap, and the endocap. Then you have your seed. If you don't call it seed, you call it ovule. Then your placenta. Placenta is the structure that attaches the ovule to the fruit. So you have the succulent hairs. So when you finish your drawing, add your title and include your magnification. Alright, so I'll be discussing specimen G and H together. Now, G is raw egg of domestic fowl, and specimen H is test tube containing 3 ml of water. And basically, what you'll be asked to do here is a type of chemical test to test for protein. Now, if you check the list of things your teacher was asked to produce or provide, is sodium hydroxide and 1% copper sulfate. And these reagents are used to test for protein. Now, this is what is going to happen. You are going to be told to put at least 3 ml of the albumin, the egg white, in a test tube. Then specimen H, which will already contain 3 ml of water. So you will be asked to put some drop of sodium hydroxide in the both test tube. That's the test tube containing the water and that one containing the albumin. You'll also be asked to put some drops of copper sulfate in the test tubes as well, both in the one with water and the one with the albumin. And you'll be asked to record the color changes, to record your results in your table. So you know your table, we have the test, the observation, and inference. Now the one with water will remain the same, will have, I think, a light blue color, while the one with albumin, the test tube with albumin, will change color to dark blue or violet color. That signifies the presence of protein. So that's what this particular specimen is all about. Specimen J is longitudinal section of boiled egg. So they can ask you the parts and functions of this specimen. Now you have the albumin. The albumin, which is the major proteinous part, it supplies protein. The yolk provides food for the embryonic growth and the embryo. The shell protects and gives shape. It also helps in gaseous exchange. The shell membrane allows gaseous exchange as well. If you're asked to draw the specimen, this is what your drawing should look like. The air space should be there, the egg whites, which is the albumin, should be there, your yolk should be there, your shell membrane, and your shell itself. So this is what the drawing looks like. So this is the review of the specimen for 2025 biology WAEC.